Thank you so much, Dr. Orchinik.
You're welcome.
So we had some questions off Twitter, just to clarify. So muscle wasting, is that related to stress?
Muscle wasting is, yes, absolutely related to stress. So the stress phenotype actually produces a lot of abdominal fat and muscle wasting. So people that are chronically stressed or produce too much stress hormone show that phenotype. The reason why there's muscle wasting is because glucocorticoids increase the glucose concentrations in the blood. And one way to do that is to break down muscle and use those metabolites to produce glucose.
So is it ever possible to look at somebody and kind of diagnose with your eyes what might be going on?
Usually it's extreme, but if you look at people who have, for example, brain inflammation for one reason or another, and they've been put on high doses of glucocorticoids, cortisol-like compounds, they will show this phenotype of a puffy face, muscle wasting, and abdominal fat. So, it's a lot easier to see it when there's pharmacological intervention.
One of the things we wanted to ask you is where, I guess, during your career has stress surprised you, and where can it surprise us? Where do you not necessarily expect to find it related to things?
So what is not stressful?
Oh no, what is stressful when perhaps we don't expect it to be? There's a concept called eustress. And what that says is essentially that any sort of engaging activity produces some degree of stress response. So anything where we're really activated and aroused will produce some sort of stress response. It's not what you'd see with a real threat, but we call this eustress, and it's just part of the idea that life is stressful. I mean, that's what, one of the characteristics of life is irritability. That's what I learned in intro biology. But now I think, one of the characteristics of life is that experiencing stressors. Life is stressful, and it's not always an unpleasant experience.
So as you've been teaching for the number of years that you have, and you talk about this with your students, do you find that there's particular topics or overarching concepts that they just have a really hard time grasping or either applying in the real world?
Well, from a teaching perspective, I think the students really struggle with negative feedback. It's very difficult to get a really solid understanding of negative feedback, especially in the context of homeostasis, because we still teach physiology, the large part in the context of homeostasis. The students will tend to think that negative feedback only occurs when some variable is too high. But say if the concentrations of carbon dioxide in the blood drop too low, you'll get negative feedback. Negative feedback is to bring a variable around a level where we want it to be, some sort of optimal level. And students struggle with that concept, but it's extraordinarily important for understanding stress. And I use the example of stress because I think it's pretty clear what the consequences are of not having negative feedback when you're talking about the HPA axis.
Something that's been elusive so far in canine science is trying to find something that would be the equivalent to a pH or litmus test for stress and to be used in animal welfare. So we've used some physiological markers like cortisol, but it's not perfect. Where do you think, given your experience, we should be looking to try and find something that is a better indicator or good indicator of how our dogs are coping?
Well, so there has been, quite a bit of effort to use salivary cortisol, or in some species, fecal cortisol. And what you get from those measures Well, salivary, you get a pretty rapid response. And it's a pretty good indicator of stress responses, an increase in cortisol within a given time frame. The fecal samples, you're really looking at the cortisol levels over the course of a day. But the challenge is to remember that there are these daily rhythms of cortisol. And not only that, there are, this is work from Stafford Lightman's group in Bristol, there's an oscillation of cortisol, or corticosterone is what he's worked on, that's beneath this stress response. So instead of a stress response being nice like this, or basal levels being nice like this, there are constant oscillations. And these oscillations may be what are leading to some difficulties in analyzing cortisol levels or corticosterone levels. So if we can get a way to track these oscillations in addition to these markers like salivary cortisol where it's not going to show, I think that would be helpful.
And as we're learning more about salivary cortisol in dogs as well, we're finding more about how personality factors, there's such huge variability between individual dogs. So it's not like the same value, the same metric from one dog means the same to the other. So say you had a level of 0.3, that's not necessarily indicating the same response from one dog to another because their average, normal, everyday levels may vary hugely between the two.
Right, so the average will miss a lot of individual differences, give you a lot of variance. We see that in all the stress research. So can I ask, are there breed differences in stress responsiveness?
That's an interesting question. I'm part of a collaboration currently doing a meta-analysis looking at salivary cortisol in dogs. And we have pulled data from approximately 6 litres, I don't know what that is in US terms, of saliva. That's a lot, thousands of samples of dogs.
6 liters of saliva?
Yeah, because
it's a nice thought.
pulled the raw data from 30 studies and pulled it together to run a meta-analysis. So we're finding more about what the average is for dogs because we didn't even know that.
And it's exciting because we're going to get to talk about this all day. There'll be a lot of saliva with us today, a lot. And
So I just wonder if there are epigenetic modifications in dogs that lead to different responsiveness to stress that or produce the epigenetic modifications due to their rearing conditions.
The good news is we've got all day. So we're going to have to leave it there for now, and we'll be back after a short break, starting again at 10:15 Pacific time. So we'll see you again soon.